Hello and welcome to the 8th Longevity Snapshot, where we will take a look at how Klotho gene therapy can increase the lifespan and function of mice. My name is Chris Linnell, and I am the founder and president of the Canadian Longevity Association, Canada's premier longevity nonprofit. If you'd like to find out more about what we do and how you can support us, then please check out the links below this video, where you can also find a link to the study being reviewed here. In addition, if you enjoyed this video, then please give us a like and subscribe to our channel. Klotho was one of the first longevity genes found in mice back in the 1990s, whereby increasing its expression lengthened mouse lifespan while knocking it out reduced it. Its name comes from Greek mythology, where Klotho was one of the three fates which governed our lifespan. She was the one who spun out the thread of life. In humans, lower levels of Klotho have been linked to various diseases, including kidney disease and increased mortality. Klotho comes in three forms, and it is the secreted form which we are looking at here. If ingested, it gets broken down in our stomach, and it is too big a protein to cross the blood-brain barrier. Therefore, at present, gene therapy is the only way to increase its levels, and that is what is employed here. This study was the first to test a single injection of Klotho gene therapy in mice on their lifespan. So let's take a look at what they found. Starting with the study design, 96 mice were used, both male and female, and they were divided into three groups. They were aged naturally, and at 6 months, two of the groups were injected, one with a sham injection of gene therapy, and the other one with Klotho using a viral vector. 6 months is the equivalent of a human in their 20s. The third group were injected with Klotho gene therapy at 12 months, which is the equivalent of someone in the early 40s. They were then tested at 24 months before living out their remaining lifespan. 24 months is the equivalent of a human in their late 60s. In all of these figures, black represents the control, light blue represents getting Klotho at 6 months, and dark blue at 12 months. Looking at the results, starting with body weight in B, with the dotted line showing 6 months and 12 months when they were given Klotho. And the control mice weight peaked at 14 months. The 6-month Klotho mice peaked at 15 months, and the 12-month Klotho mice peaked at 20 months and seemed to maintain their weight much better. In terms of median survival, the control mice lived for 24.6 months. The 6-month Klotho mice lived for 25.8 months, while the 12-month mice lived for a median of 28.3 months, which was a 15% increase over the control. And you can see the survival curves on the right here, with the dark blue line being pushed out quite a bit further to the right. Now, the authors also talked about total longevity, which I think refers to average as opposed to median. And here, the control mice lived to 26.3 months on average. The 6-month mice lived to 29.8 months, and the 12-month mice lived to 31.8 months, which is a 19.7 increase compared to the control. Now, these lifespan results are only in the male mice because the female mice had a very high incidence of ulcerative dermatitis as well as anal bleeding, and a lot of them had to be euthanized. This was seen across all three groups, and the authors think that this was not aging related. Therefore, these results sort of weren't incorporated, and therefore they couldn't calculate the lifespan of the female mice, unfortunately. However, the other tests were done in both sexes of mice. Next, they looked at how much Klotho was increased in the mice, starting with DNA expression in the liver. And as you can see, both in male and in female mice, levels increased quite considerably, especially in the 12-month mice. The same was seen with circulating levels in the blood, with once again the 12-month mice showing the highest levels. Also, it appears that Klotho increased more in the male mice as opposed to the female mice, as can be seen by the higher dark blue bars here and here compared to the female ones. Next, they tested physical function. Unfortunately, this is a journal pre-proof, so some of the headings are blocked off, but this has been peer-reviewed. It just needs some copy editing done and the layout sorted out. And they started off with a rotarod test in A, with male on the left and female mice on the right. And a rotarod test is when mice are placed on a rotating rod, which gradually increases in speed, and then the speed at which they fall off is measured. And these levels are rotations per minute. There was a slight increase for the male mice, though this did not reach significance. However, it did reach significance for the female mice, especially the 6-month mice. The second test in B was a horizontal bar where mice were left hanging with their forelimbs off a small beam, and they were timed to see how long it took before they fell off. Here, both the male and female mice saw an improvement, especially the 12-month-old mice, which reached significance, as can be seen by the star on top here, with more stars meeting greater statistical significance. Next, they looked at grip strength, where mice are sort of cling onto a wire while their tails are pulled, and the force at which they let go is measured. Now, this one is a little bit confusing. They were tested 3 times, and in the text, the authors say that the male mice saw a significant increase, but I can't really see it from the bars here. I mean, for the 6-month mice, there's an increase denoted by these sort of 3 stars up top here, but the bars are lower than the gray control bar, so I'm not sure what they're really talking about here. For the female mice, only in the first test did the 6-month mice surpass that of the controls. So, as I say, a little bit of a discrepancy between the text and this figure. So maybe that will be sorted out in the final version. Then they looked at muscle fiber size from 24-month-old mice. So a subset of the mice at 24 months were euthanized to be analyzed further. So here you can see the cross sections, and according to the bar chart on the right here, there was an increase in fiber size, though it did not reach significance. However, looking at fibrosis, denoted by the red staining in these cross sections here, for the 12-month-old mice, there was a significant reduction in fibrosis. Next, they looked at muscle regeneration. And they took a muscle tissue sample from 24-month mice and grafted them into 3-month-old mice, which have much greater regenerative potential. And you can see a cross-section of those muscle grafts here at the top, and the eyeball test seems to show that the 6 and 12-month-old mice had a greater number of muscle cells than the control mice after a few weeks. And in terms of the muscle fiber size here in B, both the 6 and 12-month-old mice had greater muscle fiber sizes, with the 6-month-old mice performing best. The control mice had a lower peak than either the 6-month-old mice or the 12-month-old mice, as you can see here. Then they looked at markers of cellular proliferation, with Ki67 being that marker. And as you can see in 2 different tissues, and Ki67 is such a marker, and connected with 2 other sort of markers, levels of Ki67 increased in both sets of Klotho given mice, indicating that Klotho gives much greater regenerative potential. Next, they looked at Klotho's effect on bones. And here behind this banner, we have mineral density, bone volume, bone perimeter, cross-sectional thickness, bone volume to tissue volume ratio, trabecular number, trabecular thickness, and trabecular space. Now, trabecular is the thicker end of our longer bones. If you sort of imagine a bone, it's sort of thin and then bulges on both ends. Those bulges are the trabecular segment of the bone. And although a lot of these did not reach significance, there did seem to be an overall improvement, especially for the 6-month-old female mice. Now, female mice, just like humans, suffer greater bone loss than male mice with age. The improvement can be better seen with these images here, showing an internal 3D image. And you can see here with the control mice how the female mice had much sort of less bone than the male ones. However, the 6-month-old female mice and the 12-month-old male mice had much higher levels of bone. Looking at the expressions of genes associated with bone growth, there was an improvement compared to the controls, with a greater improvement in females. Only FGF23 saw a decline in females. Then they looked at the brain, or more specifically, the hippocampus. And once again, the banner blocks it off, but on the left here, in these cross sections of the hippocampus, we have the control mice on the left, the 6-month mice in the middle, and the 12-month mice on the right-hand side. And in these bar graphs, it's the men on the left and the females on the right once again. Now, the authors don't really sort of explain this, apart from saying that these are all cellular markers important for the correct function of the central nervous system. And as you can see, especially for the 12-month mice, they all had higher levels of these markers than the controls, though this does not appear to be the case with the 6-month-old mice. The same goes with the second set of markers here in B, though not to the same extent. They then looked at neurogenesis, and while levels of that proliferative marker Ki67 wasn't higher, DCX, which are immature neurons, increased in the 12-month-old mice. And the thickness of the granular cells layer was higher in both sets of Klotho mice, though this did not reach significance. Finally, they looked at the transcriptomic profile in the hippocampus, and they found 3 distinct clusters of variants between green 5-month-old control mice, old control mice, and old Klotho-treated mice. And looking at this Venn diagram on the right, which shows differentially expressed genes between either young control mice, old control mice, or young control mice and old Klotho-given mice, there was a much greater number of differentially expressed genes between the young and old control mice compared to the young control mice and the old Klotho-given mice, which indicates that the old Klotho-given mice were much closer to the young control mice in terms of gene expression. Lastly, they looked at what those different pathways were, and they found that compared to the control mice, the Klotho-treated animals presented increased induction of phagocytosis. So that's immune cells which engulf sort of bacteria and viruses, increased immune cell activation, and a decrease in pro-inflammatory signals. Finishing off with the graphical abstract to summarize, we saw increased longevity, up to 20% in the Klotho-given mice, enhanced physical condition, I guess that's more physical function. If the mice look better, they didn't mention it in the text or provide any photos. You can see, I guess, the hanging beam test here, and so showing improved muscle regenerative capacity, improved bone microstructure, improved histological markers in the central nervous system, reduced inflammation signaling, and promotion of phagocytosis. Now, a few last things to mention. For some of the tests, you might have noticed that the 12-month-old mice performed better than the 6-month-old mice. And the authors think that as Klotho is more expressed in the liver, and the liver has a sort of a cellular turnover, by the time the mice were tested at 24 months, there was less Klotho in their systems, which actually the serum Klotho levels seem to indicate that as well. Secondly, they obviously need to retest longevity in female mice, and hopefully they won't have those sort of disease issues they had with them, which shows the sometimes random nature of experiments where weird things can happen. They should also test out what happens when Klotho gene therapy is given to even older mice, as the 12-month-old Klotho mice are only the equivalent of humans in their early 40s. The question is, what effects would it have if it were given to, say, 24-month-old mice, the equivalent of humans in their late 60s? But this is a very intriguing result. As I mentioned at the beginning, this was the first time that a single injection of Klotho gene therapy was given to normal mice. And it's great to see the significant increase in life and health span, especially as Klotho gene therapy is hopefully soon to be tested out in humans. Doing a bit of research on the web, I found that a company called Klotho Neurosciences is hoping to start human testing in the 4th quarter of 2025. So with a bit of luck, we might get some human results fairly soon. And that marks the end of another longevity snapshot. If you found it interesting, then please give us a like and subscribe to our channel. So until next time, goodbye.